afternoon. My name is Genoa Blankenship, and we want to welcome you to today's EMSL Learn webinar, where we will be discussing the highlighted capabilities that will be available through the fiscal year 2024 Exploratory Call. And while you're here, we want to invite you to next month's, or actually, I guess we're technically in June 28th's webinar, where we will be discussing EMSL's open on-demand software that is specifically to help you have remote access to our computing resources. We will send more information out through our newsletters. So if you are not following EMSL as part of our subscription, we invite you to join us and also to follow EMSL's social media channels for more information. We will go ahead and drop some links in the chat so that you have access to all of that information. Today, we have 4 speakers who will be talking about our different topics. We're going to start with EMSL Project Manager, Samantha Miller, who will provide details on the exploratory call itself and on deadlines and proposal call requirements. Following will be EMSL biologist, Beliaev, who will discuss EMSL capabilities in single-cell biology, cell-free protein expression for structural biology, stable isotope labeling using multi-omics analysis, and activity-based probes to explore enzyme function and biogeochemical pathways. Earth scientist Odeta Qafoku will detail EMSL capabilities available for imaging soil organic matter, as well as measuring the spatiotemporal distribution of roots and root exudates, metabolites, and other elements. And finally, we'll end with computational scientist Satish Karra, who will walk through EMSL's tools for exploratory and statistical data analysis, as well as EMSL's capabilities for harnessing artificial intelligence and machine learning methods to mine information from 2-dimensional chemical maps, process multimodal data, help co-register spatial data, and expand 2D chemical information into a 3D space. And with that very long explanation, I'm going to toss it over to Samantha Miller to begin. And please hang around the last 15 minutes because we will have a Q&A session, and we invite you to put your questions in the chat and our speakers will answer at the end. So with that, I'll let Samantha begin.
Thank you, Genoa. So I want to start by giving a bit of an overview about the exploratory research call. So this call differs from some of EMSL's other calls in that it focuses on emerging capabilities or suites of capabilities rather than on science topics. A full list of these capabilities can be found in the call announcement for FY24, and you'll hear more about them later through this webinar. The highlighted capabilities need to make up at least 30% of the research effort in terms of sample numbers or requested hours. I also want to point out that these are relatively short projects, only 9 months long, and they start January 1st and end September 30th of 2024. Last year, EMSL successfully piloted using a dual anonymous peer review process, and we will be continuing that again for the FY24 exploratory research call. For this, it means that peer reviewers evaluate the proposals without knowledge of who the team members are. This requires a little bit of work from the submitters in that they must write their full proposal in an anonymized format without any references to that could give away the team members. Sections of the full proposal that need to be anonymous include the title, abstract, project description, and references in the cited in the text. You can find detailed guidance including examples of how to write anonymized text on the EMSL website. I also want to point out about the dual anonymous process that the letter of intent or the LOI is not required to be anonymous. And this is because the LOIs are not evaluated by the peer review panels. The LOI submission is already open for the exploratory call, and LOIs will be due on July 6th. We will send out decisions for LOIs on July 28th with whether or not you're invited to submit a full proposal. Full proposal submission will open on August 1st, and full proposals are due August 31st. Final award notifications will go out November 30th. Next up, I want to highlight some important changes from past calls. All call guidance for the FY24 exploratory call has been updated to improve clarity and consistency. So it's very important that if you are planning to submit to this call that you review the website and the guidance carefully. A few key important changes include that we will be requiring a standardized template for LOI and full proposed research submissions. For the LOI template, we are moving away from a 2-page written description of the research to a form with guided questions and shorter word limits. Both the LOI template and the full proposed research template can be downloaded from the EMSL website. For the full proposed research template, this is not too different from what we've done in previous years. The project description is still 4 pages long, and there's still several required and optional appendices. What has changed is that we have put all of these pieces into a single document that users can submit. And we hope that this will make the process easier for users and eliminate some of the past problems that were experienced, such as forgetting an appendix or trying to upload multiple documents to the EMSL user portal. And with that, I'd just like to say that I'd be happy to answer questions at the end during the Q&A session, and you can also reach out offline to user program services if you have questions about the proposal submission process. Next, I will turn it over to Alex to talk about capabilities in the functional and systems biology science area.
Okay, thank you, Samantha. Hi, everybody. I am Alex Beliaev, the IRP lead for cell signaling and communications. And today we'll talk about the call topics that are aligned with the EMSL's functional and systems biology science area. This area focuses on revealing the connections between protein structure and function, biochemical pathways, and complex phenotypic responses. And our strategy, our approaches incorporate multi-scale and multimodal experiments, reconstruction of metabolic pathways, computational modeling to reveal the new strategies and ways how we can design organisms for biofuels and bio-based products, as well as to ultimately understand the complexities of nutrient and energy cycling within cells and their immediate environment. The scientific and technical expertise in FSB is clustered around 3 integrated research platforms. These are structural biology, biomolecular pathways, and cell signaling and communication. And these IRPs serve as primary interfaces for users to connect with EMSL science staff and capabilities. So in the next 10 minutes, I'm going to go over the FSB topics for this year's call and highlight the specific resources within each of the IRPs that are targeted in this year's call. Okay, so our first IRP, structural biology, focuses on examining the assembly, structure, and function of proteins and protein complexes at the nanoscale. And this year, we're seeking proposals that specifically emphasize the utilization of cell-free expression systems for structural biology, pathway engineering, and biomolecular analyses that contribute to functional annotation of proteins, especially those that are highly conserved across the fungal kingdom. So this capability in EMSL is stewarded by James Evans and Irina Novikova, who initially developed the cell-free pipeline using the wheat germ-based expression system. We primarily use the WeGerm workflows for most of our DOE-BR samples, but we also have the E. coli and human TXCL lysates that perform both transcription and translation to really allow us access to different post-translational modifications. This enables expression of bacterial, fungal, algal, plant, as well as human genes in different scales. Most work here, though, is done at the mini scale, which is used to evaluate whether target expresses or if any additives are needed to make the protein more soluble. The membrane proteins can also be expressed with liposomes, nanodiscs, and detergents. So the expression conditions are optimized. We can scale up using the Protemus robotic protein synthesizer available at EMSL. And we can use two scales: midi scale, which can synthesize up to 24 proteins without purification, which is ideal for functional characterization and annotation. And we also have the maxi scale, which is used to conduct structural studies, and we can purify and synthesize and simultaneously purify up to 6 proteins. There are different entry points here. We can work with users to generate the clone or work with users that already have clones if they meet our specifications. There are additional details also about the cell-free expression pipeline and protein structure function analyses, and these can be found in the recorded webinar on the EMSL website. Also, as I mentioned earlier, this year, we're looking for proposals that will contribute to the 1,000 fungal proteins project, which aims to associate structure and function with previously uncharacterized or poorly characterized proteins that are highly conserved across the fungal kingdom. This project, which is done in collaboration with JGI, leverages EMSL capabilities in cell-free expression, native mass spectrometry, NMR, cryo-EM, as well as high-performance computing. The overarching goal here is to provide new characterization from the structure of small molecule metabolites and link that to the structure and function of annotated proteins, as well as visualize and analyze the micromolecular complexes in situ within the fungal cell environment. Here, as I mentioned before, we can engage with users at various points of the pipeline, including users that already have highly purified proteins or metabolites, and those who only have a gene sequence and would like to utilize our computational and experimental workflow. The pilot here has already begun, and any users that are interested should contact Scott Baker and James Evans about how to access the resources through this year's exploratory call. Moving on to the biomolecular pathways IRP, which we are seeking this year topics that emphasize experiments utilizing stable isotope labeling and multi-omics to study the flux of carbon and nutrients through natural and engineered systems, and enable fundamental and applied studies. The supporting capabilities that I would like to highlight here encompass a broad range of mass spectrometry and NMR instrumentation. For biomolecular pathway characterization, we measure label incorporation to different organisms and metabolites to carry out metabolic flux analysis and trace carbon and nutrient partitioning across central and secondary metabolic pathways across different microbe species, and identify identify nutrient partitioning across different pathways as well as different molecules. We can detect biomolecule speciation as well as it relates to percent enrichment, as well as molecular position to measure quantities of newly synthesized metabolites, proteins, lipids, transcripts, and that enables stable isotope probing techniques. Here, mass spec imaging can also be deployed to generate spatial, qualitative, as well as quantitative molecular information to enable visual proteomic or metabolomic investigations. Finally, a suite of NMR, LCMS, and IRMS capabilities is available for tracer studies of challenging matrices that include, but are not limited to, high-resolution chemical analysis of soil organic matter, as well as solid-state NMR for analysis of samples that has little to no mobility, such as soil particles as well as cell wall material. Again, we have a webinar available on the EMSL website, and those interested in specific isotope labeling workflows, instruments, and details are welcome to take a look at it. And finally is the cell signaling and communication IRP, which investigates dynamic interactions and trafficking of molecular signals between cells, populations, and communities to understand the interrelationships between organisms and their response to the environment. This year, we have several topics which center on studies that move beyond species-resolved bulk measurements by improving the sensitivity and specificity of our multi-omics platforms, as well as chemical biology approaches to characterize metabolite enzyme interactions and visualize spatial enzymatic maps. In regard to supporting capabilities in single-cell biology, we have been developing new cell separation, molecular labeling, and signal amplification strategies for quantifying transcripts and proteins at a single cell level to reveal genetic diversity as well as functional heterogeneity within populations. Workflows that are of interest to us in further developing include nanodroplet deposition and processing for simultaneous transcript and protein measurements, integration of this platform as well with nanopods mass spectrometry. RNA-seq enables quantification of both mRNA and protein expression from the same single cells, also revealing the interplay between translational and transcriptional processes. We're also extending the utility of single-cell transcriptomics to prokaryotes, where the smaller cell size and genetic heterogeneity hinder the application of deep sequencing technologies. And to address these challenges, we are adopting, in collaboration with University of Washington and Institute for Systems Biology, a recently developed high-throughput split-pool ligation barcoding transcriptomics that can detect and resolve microenvironment-specific variations in mRNA and apply that to complex multi-species systems. In the chemical biology area, we are interested in capabilities employing activity-based probes, which are small chemical compounds. You could see the design of the chemical probes on that slide here. The combinatorial approach to ABP design includes incorporation of reactive element that binds to the target protein target, the recognition element that provides specificity, and the reporter element that allows downstream detection, and it enables application of multimodal measurement strategies. For example, here, as you could see on that slide, LCMS can be used to determine relative abundance and identify probe-labeled proteins, while fluorescent microscopy can allow spatial mapping of probe-bound proteins in whole cells. The ability to attach different fluorescent markers also opens an opportunity to isolate and identify enriched microbial subpopulations using FACS and use that for downstream functional as well as omics measurements. EMSL has already a large library of ABPs available for profiling of different protein activities that include cysteine and serine proteases, as well as hydrolase activity proteins. Additional probes also that feature novel chemistries can be developed and tested in collaboration with the users for biochemical function or substrate of interest. We're very interested in growing our ABP pipeline and workflows and develop new cross-cutting capabilities that not only support the FSB science, but also provide linkages to other areas, in particular to user projects within the environmental transformation thrust. If you have any questions with regard to the activity-based probes, please contact Sankar Krishnamurthy, who is the scientist at EMSL, leads the development of activity-based workflows. And with that, I'm going to wrap up my part of the webinar and introduce the next speaker, Odeta Qafoku, who will talk about the highlighted capabilities for the rhizosphere processes area. Thank you.
Hello, I hope everybody can hear me.
Yes, go ahead.
Okay, thank you. So I'm Odeta Qafoku, and I'm Earth Scientist in EMSL, and I will continue our discussion on exploratory proposal calls. And this time I will talk about exploratory proposal calls that apply to rhizosphere processing and cis and biogeochemical transformations. And in the environment, we know that the rhizosphere processes are very complex, as are shown here by a number of processes in the circles. They include interaction of roots with microbes and with minerals, and exchanges of nutrients, and also all that is perturbed by environmental factors. And the aim of our exploratory proposal calls is to advance understanding of these processes and mechanisms. And specifically, we are calling proposals that are interested in understanding the spatial temporal distribution of the root metabolites, the microbial exudates, carbon and nutrient exchanges, and also organic matter stabilization in the environment. So for that, we are seeking proposals that are going to use some of our highlighted capabilities that are novel and emerging capabilities in EMSL. And those capabilities that I will start first are called terraforms. We have a new name for the synthetic soil habitats. And these capabilities have been developed in the last 2 years in our facility. And they are very, they can be customized, and they are also versatile. The first capability I would like to talk first is the rhizochip, also that it stands for rhizosphere on a chip, and it is shown by this. It is shown by these pictures here, right? Is shown that by this picture in here, and also by a couple of other images in here. This device is targeting soil properties, physical soil properties, such as soil pores and soil structure, and how that impact the root growth. In addition to being easy to be customized based on the texture and the structure of the soil of your interest, this device can also be is versatile, because it can be we can control the environmental factors as we can control specifically humidity or carbon CO2 concentration, temperature, light, and we can see how all these factors can impact in these not only the root growth, but also in the metabolite production. And to do that, I'm showing here an example how this capability can be integrated with other chemical imaging capabilities that is shown here, for example, multi-mass spec, to indicate how this spatial distribution of metabolites is occurring. And also it can be coupled with fluorescence microscopy, which is shown here in this figure down, in this image down, coupled with fluorescence to see where the microbial community is in relation to the root plant root. And for any questions about this capability, and if you would like to use this in the exploratory proposal call, please contact Jade of REC. The next capability that I would like to show is another terraform, and it is called synthetic micromodal. This device is similar to the first device that I showed with respect to the targeting soil pores and porosity and structure, but is also unique in the fact that we can also target soil mineralogy using this device. And for that reason, we can dope this device with one single mineral or with a mixture of minerals and study abiotic processes. Here, for example, I'm showing how this particular device that is a highlighted capability for the exploratory proposal, can be coupled with with SIMS, SIMS or X-ray fluorescence spectroscopy, microscopy for understanding nutrient transport or organic carbon transport. Also, it can be coupled, as you can shown here down, with multi-MS to look at the type of organic carbon that is being released. This device is also very versatile because in addition to being used for abiotic processes, as is shown in this, actually here is targeting fungi mineral interaction, it can also be used as a microfluidics device, and it can address biotic processes, those that can be driven by the mineral liquid interaction. It can, for example, it can study the adsorption processes, it can study the redox processes, and those all that can be controlled by the fluid residence time or the pore size distribution. Any questions about this device? The Terraform device can be directed to Aruna Bhattacharya, and she will be able to tell you more about that specific application of this device. The next highlighted capability for the next highlighted capability that I would like to talk here is the X-ray computed tomography. This is another instrumentation capability that we have here in EMSL, that we have used it for looking at the root architecture. One of the strengths of this capability is that it's not destructive, and it can look at that distribution of the roots below ground, which gives us insight of what the environmental conditions are allowing this growth. In addition to just looking at the architecture itself, this device can also be, this capability can also be integrated with omics extraction, as it is shown in this series of images, where you can look, you can investigate the microbial distribution, the metabolite distribution across the roots. And for this specific work of combining the metabolite distribution with the root architecture, please contact Pupudu Handakumbura. She is an expert of these techniques. The XCT also can be coupled with another number of techniques and chemical imaging techniques that we have in here in EMSL to address not only what is happening in the plant root system, but also what is happening in the surrounding environment. For example, it can be coupled with nanosims. In this case, we'd like to have isotopically enriched the system to be studied by this instrumentation that we have available here. And the nanosims can not only look at the root distribution within the root, carbon distribution within the root, but also distribution around the root in the nearby environment, around the microbial network, or nearby soil environment. Also, laser ablation mass spec can be integrated with XCT to study the nutrient gradient around the root system. If you want more information about this, how these devices can be applied, integrated with X-ray computed tomography, or alone as a highlighted capabilities, please look at a webinar that has been recorded by EMSL team and is available to you for view. Also, other capabilities that we can attach to the X-ray computed tomography for understanding processes in nearby environment are, again, multi-mass spec to look at the carbon concentration around the root, and electron microscopy for looking at morphology around the root, and also for looking at hotspots that are forming around the roots and around that environment, which can give us insight about organic carbon destabilization or stabilization. So when I talk about the organic carbon stabilization and stabilization, I also want to highlight here a number of electron microscopy, high resolution electron microscopy techniques that we have that can give much more in detail what is happening at the mineral microbe interface and how we can identify formation of some biofilm, for example, as is shown into these two images here, and which actually serve as a source for accumulation of other minerals and for aggregation, early stages of aggregation. Electron microscopy has also a different type of application that's called focus ion beam application that can be used to do a cross-section of the microbe mineral interaction and look at what is happening just below the surface with respect to bioweathering or with respect to elemental distribution, which in that case, it can be understood by coupling another capability which we have here, which is atomic probe tomography that gives a three-dimensional distribution of elemental and nutrients in that area of interest. All the capabilities are stewarded by Shuta, Alice, and Jennifer and myself. So please contact every one of us if you have further questions about these processes. And I did mention that the XCT is used for getting the root architecture, but XCT can also be used very efficiently for understanding also soil structure and soil physical properties distribution. And this is a video that is an image of a soil structure, and those structures can be microaggregates or macroaggregates, which are larger, or soil cores, and it will give us a good distribution of the pores, whether they are connected, which are shown here in the blue, or whether they are not connected, which is shown in the purple. And the way these pores are distributed within a soil column or soil structure is important, again, for plant growth and for the nutrient movement. One of the capabilities that we are recently developing is taking the 3D physical characterization and combining it with 2D chemical mapping. I mentioned earlier on that this XCT is a non-destructive technique, but to get a biogeochemical characterization of the core, we have to do some physical manipulations of the aggregates in this case, and form some thin section, and then take these thin sections for characterization with electron microscopy, energy dispersive microscopy. And what we get now, we get this chemical distribution on all these thin sections, and this is our first step into getting this a coupling of 3D soil physical characterization with 2D chemical distribution. And in this top 3 slides here, I'm showing the elemental distribution with respect to these aggregates that I just showed, the XCT 3D mapping. And this distribution can is very telling to what exactly is happening around pore space and how that pore space can drive a certain reaction. And to get that, we can get even higher resolution to understand the chemistry around the pore space. Here is driven by iron chemistry, or where the organic carbon is with respect to the minerals, as is shown here, is entrapped within a mineral grain. All this characterization can also be coupled with other imaging capabilities, as is MALDI that is shown on the right, which can tell us nature of the organic carbon and how that spatial distribution is with respect to the location of the roots, etc. I will also mention very briefly that if you are interested in the iron chemistry, please consider adding the Moss-Bauer spectroscopy as a capability that can be attached to the highlighted capabilities, as it can give some great information about the iron chemistry in the sample of interest. For that, please contact Ravi Kukadapu. And finally, I would say that to go from 3D physical distribution to 3D biogeochemical distribution, we have to couple it with the machine learning and artificial intelligence. And that is a next step that EMSL is working very hard about, and it's going to be part of the next speaker conversation. And with that, I hope that you have a good view what highlighted capabilities are proposed for the rhizosphere and biogeochemical transformation, and how they can be coupled with other imaging techniques. And with this, I will give this to Satish to talk more about machine learning and AI.
Thank you, Odeta. Okay, good afternoon, everyone. My name is Satish Karra. I am a computational scientist at EMSL. And in this part of my presentation, I will highlight the capabilities and call topics of interest as part of computing, analytics, and modeling science area of EMSL. All right, so first aspect is that we are seeking AI and ML methods to mine information from EMSL instrument data. So what I'm highlighting here is we have a series of experimental instruments, including the ones that Odeta and Alex have highlighted in the previous parts of the presentation. So we are seeking ML and AI techniques that will help us mine information from these instruments, use multimodal data streams towards scientific discovery, help co-register spatial data from these instruments, and so on. Now, another example of AI ML that's of interest is the mapping of, for example, 2D chemical information to 3D. And this is something Odeta highlighted in the previous part. And I'm just showing you as an example where one of the aspects is to use the information from 2D geochemical electron microscopy images and map it to 3D pore structures using AI or ML techniques. And the third aspect of bringing AI ML into our workflows is we are seeking testing of AI ML methods for molecular simulations. EMSL has a strong expertise and suite of capabilities in the area of molecular modeling, including NWChem toolkit. And as an example of AI ML application could be the development of, for instance, fast running emulators based on machine learning and AI. So on this topic and these capabilities, if you have any questions, please do not hesitate to reach out to me. Moving on, I'd like to highlight the next capability that's of interest as part of this call is, and this is called MAP, which is multi-omics analysis portal. And MAP is a web-based tool consisting of a suite of analysis and visualization tools, primarily for multi-omics data coming from various EMSL instruments. And so as part of this call, we are seeking for exploratory and statistical methods that will use this capability of MAP, and that's of interest. So some of the data I've highlighted that is supported by MAP currently, including metabolomics from NMR and LC/GC mass spectroscopy instruments, Fourier transform, ion cyclotron resonance mass spectroscopy, LCMS, lipidomics, etc. If you have any questions regarding MAP or this area, please reach out to Kelly Stratton. I also would like to highlight next the computing resource at EMSL that users can take advantage. In this case, the development of, say, the computational methods related to AI, ML, or the multi-omics data analysis. This includes Tahoma, which is our heterogeneous scientific computing facility. And I'll quickly touch upon it in the next slide. We also have Aurora, which does the archiving of the data. So if you are using data either coming from EMSL instruments or your own data for building AI, ML techniques, this can be archived on Aurora. And thirdly, we have cloud computing infrastructure based on Razor clusters. For example, MAP, which I highlighted in the previous slide, is hosted on one of our Razor clusters. So when you are proposing something around MAP, this is a capability that might be of interest. And then, as I said, the scientific computing, I just wanted to quickly go into the details of the scientific computing heterogeneous machine Tahoma. So we have 184 nodes, and 24 of these nodes are specifically dedicated for AI, ML, and these are NVIDIA Tesla GPU for AI and ML that users can use for the development of computational methods. And if you have any questions with regard to the computing resources at EMSL, please reach out to me. With that, I will end my part of the presentation, and we will move on to Q&A session next.
All right, great. Thanks, Satish. I'll invite the speakers to come back online or turn your videos on. We do have some questions, and I just want to remind our attendees that if you have questions for any of the presenters, go ahead and put them in the chat. So first question is for Samantha. What do I do if I don't know which EMSL resources to request?
Yeah, that's a great question. First, I would recommend reaching out to one of the scientific points of contacts that were listed throughout this webinar and are also listed on the EMSL website. Those are the people who can help you develop the technical feasibility of your proposal and point you in the right direction for what resources are appropriate. If you can't find the appropriate contact from the website or from this webinar, you can also reach out to EMSL User Program Services, and we're happy to find the right contact for you.
Great. Thank you, Samantha. Next question is for Satish. Satish, are you interested in topics such as running scientific computing workflows to cloud, high performance computing, edge with resources, resource management tools such as, I'm probably going to butcher this, Kubernetes?
Kubernetes.
Okay.
Yeah.
What about, there's more to it. What about big data management and data reduction with lossless and lossy compression techniques or loss compression techniques?
Okay. Thank you, Genoa. So I'll just go with the first part. With regards to Kubernetes, we do support Kubernetes on our facilities. So, for example, the Razor clusters that I highlighted as part of the cloud computing infrastructure, we do have Kubernetes on that. So if there is specific scientific computing workflows, which are including AI, ML techniques, we can support Kubernetes. Now, moving on to the lossless compression techniques. Again, as part of this call, big data management and data reduction specifically, again, I'm not 100% sure on what aspects you're interested in, but anything that supports AI, ML techniques, the multimodal data streams, that is definitely of interest.
Okay. Great, thank you. Question for Alex. Alex, are there specific sample requirements for single cell transcriptomics analyses?
Sorry. Yes, there are several specific requirements that we have for the single cell transcriptomic workflows. We can work both with user supplied samples that typically are fixed in methanol, or we can also, in collaboration with the user, carry out the experiments, the growth experiments. In EMSL, the key requirement here is really the amount of cells that needs to be higher than 2 to 10 to the 6 cells per mil per experiment, as well as the cells. We are currently working only with planktonic systems that allow sample preparation using cell sorting. So these are the primary requirements. In terms of the types of the organisms, we have tested our workflows with a number of model systems that include E. coli, Bacillus, Pseudomonas, but we are willing to work with the users to optimize some of these workflows for the user supplied organisms.
Great, thank you. Odeta, a question for you. How can the elemental maps and the EDX data used for upscaling of, I think we're missing a word there. How can the elemental maps and the EDX data be used for upscaling of processes?
Yeah, that is a great question. And I think the upscaling is something that everybody wants to do. And we definitely are giving a lot of thought for that in EMSL here. One of the ways that I am looking at those data is that we can create patterns using this data, EDX data maps. Those are very rich data sets, and we will need to involve data mining to get that information out. We probably have thousands of data points that we can extract, and looking for those patterns will be the first step into the upscaling.
Okay. Another question, and this might be for Samantha, but I'll let anyone take it. Is it important or necessary to include someone from EMSL in the proposal to get funded?
That's a good question. First, I want to point out that EMSL staff shouldn't be listed as co-investigators or team members on a proposal, but I would say that you have a higher chance of having a strong proposal if you work closely with staff, because they have the most technical knowledge to help make sure your proposal is feasible with the resources that are available. And they are ones who would work on the project likely if it is funded.
Great, thank you. Satish, question for you. Do the artificial intelligence and machine learning methods have to be novel, or can they be existing, well-tested methods?
Yeah, so the novelty could be in terms of using existing methods on our data streams. So the multimodal data streams coming from EMSL instruments, the methods necessarily don't have to be, you don't have to build them from scratch. If there are existing AI ML techniques that are well tested in other areas, but if you can bring them to EMSL and integrate with our instrument data streams, that would be novel for us, and that is within the bounds.
Okay. Follow-up question for you. Will there be support from EMSL on using MAP and making any modifications if needed?
Absolutely, yeah. So we have the expertise in-house. We will provide the support that users may need with regard to MAP, and if they are bringing their own techniques in and want to integrate with MAP, we have the in-house help that we can provide with that.
Okay.
Thank you.
Great, thank you. Odeta, another question for you. What is a procedure that has been used for replicating soil properties using terraforms?
Yeah, that's also is a great question, and we are still developing these devices to represent the soil properties as closely as we can. One way that we have approached is looking at the soil structure and either by observing aggregate from that soil of interest and using that, using XCT to get the porosity and pore distribution, and that doing the 3D printing of that distribution into those devices. That's the approach that we have used so far. And I'm sure that more and more we work with these devices will improve them to represent the soil properties much better.
Great. Thank you. Alex, question for you. Does activity-based probe workflow require live cells?
No, yeah, thank you. That's, yeah, it depends on the type of the protein target and really the scientific goal of the experiment. Typically for probing enzymatic activity or discovery of new functionalities, we can use cell lysates. So that's not really live cells. We can get either the live cell pellets or extracted protein, ideally at concentrations about 1 to 2 milligrams per mil of protein. We can use also fixed cells, if fixed whole cells, if we are doing microscopy or cell sorting, and we can also, again, we can use live cells, and in that case, obviously the cells can be grown at EMSL, and we can do that in collaboration with the users to do any types of in vivo assays using activity-based probes. Thank you.
Thank you. Okay. This might be a question for Samantha. Just to confirm, EMSL staff, are they prohibited from participating in proposals?
As I said before, they can't be listed as PIs or co-PIs, but they can work with you to help develop the research plan. And there's also a space in the user portal when you're making a proposal submission to list the scientific contacts that you had at EMSL. So it's important to name staff that you worked with there because then they will likely be added to your proposal if it gets funded and participate after it's approved.
Okay. All right. Looks like we have one last question, and then I'm just going to remind folks that if you have any burning questions, please drop it in the chat before we sign off today. The last question I'm seeing is, Samantha, what tips do you have for writing a successful proposal? Or actually, anyone can answer this also.
I'll just throw out one, which is to review the guidance very carefully and make sure that you understand it well in advance to the deadline. Also make sure that you log into the EMSL user portal and make sure that you understand how the portal works and what information you will need to submit your proposal.
Great, thank you. With that, that is all the questions that we have for right now, but just a reminder that you can reach out to anyone that is on this call as a presenter or who is listed, as an instrument owner for more information. And we hope to see you in a couple of weeks on June 28th at noon for the next EMSL Learn webinar. And if you have any colleagues that missed today's webinar and are interested in submitting a letter of intent, just remind them and spread the word that we will have a recording of this webinar available usually within about a week. So thank you and have a great day.